This is the last portion of our lecture 1. Until now, we have had a brief idea about some basic terminologies and the basic concepts of biotechnology and the role of genetic engineering in recent advancement of biotechnology. We also know another term which is called recombinant DNA technology, or RDA, which is the key ingredients or the tool for such genetic manipulation of living organisms. Then we start to know about the discoveries and roles of certain scientists who have had a revolutionizing role in modern science. One of them was Louis Pasteur. And if I recall the role of Louis Pasteur, then there are two mentioned discoveries were there. The first one, Louis Pasteur invented the responsible agents for microorganisms to carry out any fermentation reaction. That means fermentation reactions does not carry out by their own or for any kind of de novo chemical changes, but the microorganism work on certain substrate in order to have food or other living energy and give us the byproducts which are helpful for us. Another discovery of Louis Pasteur was the process of pasteurization, which is another revolutionizing discovery in food science. Till now, we are using the pasteurized milk. Pasteurization is a process of applying heat in order to kill some harmful microorganisms without making any change in the texture, flavor, or taste of that certain food. And the example were given there was heating up wine to kill the bacteria or other microbes that were responsible to make the wine sour. And he did this by remaining the texture and flavor of the wine as same as before the pasteurization procedure. Now in this portion, we'll know something more about the scientific discoveries. Theory of evolution. Charles Darwin, another renowned scientist of evolutionary scientist. He hypothesized that animal population adapt their forms over time to best exploit the environment. He gave us the idea of adaptivity, that we become adapted to a certain environment either by evolutionary changes or by deliberate changes in us in order to have the best living or in order to survive. His another important statement was survival for the fittest. That means one who fits will survive. So he gave us the theory or the concept of natural selection. He traveled in the Galapagos Island and observed how the finch beaks, that means there is a bird called finch and their beak on each island were adapted to their food source. That means each of the island has the same bird finch, but they have different structure of their beaks depending on the food source. And that's why, or so this way, he get the idea about natural selection. That is, nature select the birds who are best suited in a certain environment or certain atmosphere, or nature have given us the ability to cope up with certain environment in order to survive. This is the theory of evolution given by Charles Darwin, one of the most important theory for biological science or evolutionary biology. Here is another great scientist named Gregor Mendel. Basically, he was a monk born in Austria, a very beautiful country of Europe continent. But he has given us the two basic laws or two important laws through which now we are able to know the genetic inheritable or inheritance patterns of all the living organisms. That means now we have the idea that why we have certain characteristics obtained from our mother, some from our father, and other from other relatives, and what is the proportion or percentage of our having such criteria. These two life-changing laws were named law of segregation and law of independent assortment. And I know all of you have the idea about these two laws. So let's see what he did. The lifespan of Gregor Mendel was in between 1822 to 1884. He was an Austrian monk, often considered as the founder of genetics. He gave us the idea or the basic concept about genetics, not genetic engineering. Do not be confused. Genetic engineering and genetics are two absolutely separate terms. Genetic engineering is the manipulation of in DNA or RNA level or genetic level. But genetics is what is our genetic material, what will be our genetic characteristics, and how they will nurture us and how they will be carried out in further generation. This is genetics. So in 1863, he conducted the first genetics experiment using pea plants. We all know what is a pea plant. Simple pea plant were used to conduct the experiment, but he did his study or he worked in a very persevere way. He never get depressed or get demotivated. He continued to his work in order to get the best desired observation or observable calculation. He presented his laws, the two laws of genetics. One is, and they are called Mendelian law, the law of segregation and law of independent assortment, or laws of heredity, to the Natural Science Society in Brunn, Austria. Mendel proposed that invisible internal units of information account for observable trait. Please look at the statement. He proposed that invisible internal units, and these three words refer to today's word of gene. We know that gene contain all the information for our observable traits or our phenotype. That means our eye color, hair color, our height, weight, behavior, mood swings, everything is dependent on the coding sequence within a gene. But by that time or on that time, Mendel was not sure about the term, so he referred it as an invisible internal unit and termed them as factors. The most important point, please keep in mind that gene was at that time called factors. These factors, which later became known as gene, are passed from one generation to the next. Now we know that we carry our parents' gene or our gene of our family ancestors, but at that time they used to call it factors. That means there are some factors that passes to the offspring carrying certain codes that will be observed by their phenotypes. Here we can see another scientist, Robert Koch, who investigated anthrax. Anthrax is a very common cow disease. It is also common in case of our country. We know that anthrax occur in cattle, specifically in cows, and sometimes people are also get infected for this anthrax. And the responsible agent for anthrax is Bacillus anthracis. It is a bacterial disease. You shall know it more about this type of bacteria at your or in your BTE 102 course. So Robert Koch investigated anthrax and developed techniques to view, grow, and stain microorganisms. He then photographed them, aided by Graham, Cohn, and Weigert. Three other scientists helped them to take the photograph of such microorganisms. Then studying fowl cholera, fowl means cattle type, cholera, Pasteur published his work on attenuated or weakened strains of organisms that could not cause disease but protected against severe forms of the same disease. The line within this inverted commas or the word within this inverted comma is attenuated. Attenuation means to mitigate, to lessen, or to reduce. Attenuated or weakened, that means some organisms that has been intoxicated or that has already lost their pathogenic capability or their disease causing capability. And Pasteur claimed that in one of his work that some microorganisms, when they are being attenuated or they are being intoxicated or weakened in a way that they cannot cause disease, have the ability to protect the host against that particular disease. Let me give you a practical example, such as cholera. Vibrio cholerae. Vibrio cholerae is a bacteria that is responsible for causing cholera in human. So, at one level, well, Vibrio cholerae has been attenuated in such level that they are being weakened or they have lost their ability to cause cholera. If we incorporate or we inject such weakened strains of Vibrio cholerae in a specific human, then we can see that those human or that particular human will never have cholera ever in his or her rest of the life. And this is the concept of vaccinology. For vaccine, we use attenuated, or weakened strains of microorganisms in order to get protection against that particular microbes. We'll learn more about vaccinations in BT 102. So Robert Koch describes bacterial colonies growing on potato slides, on gelatin medium, and on agar medium. Nutrient agar became standard tool for obtaining pure cultures. That means nutrient agar is certain media where we can culture some pure lines of microorganisms. Now what does it mean, pure cultures? Well, pure culture means one specific types of organism. It can be either specific bacteria such as Vibrio cholerae, Bacillus anthracis, or any other types of bacteria, but alone, not in combination with others. This is to be the most important discovery in the rise of microbiology. So, culturing microorganisms in pure form is the most important discovery in the rise of microbiology. And who did this? Robert Koch did this. Well, I repeat that these dates are not so important, but who did this and what they did? Like Robert Koch investigated anthrax. Later on, he investigated or he showed how to culture organisms within nutrient agar. And who claimed that nutrient agar, or who helped to establish nutrient agar as to become a standard tool for growing pure culture lines or pure culture microbes. These discoveries are very important for all sort of questions. Pasteur used attenuation to develop vaccines against bacterial pathogens of fowl cholera and anthrax. This was founding moment in immunology and opened new areas in the field of preventive medicine. Well, immunology is a vast term. You have 2 courses regarding immunology, so you will learn more about it in that courses. Robert Koch stated his postulates for testing whether a microbe is a causal agent of disease or not. So what Robert Koch again did, he postulated that whether a certain microorganism is responsible for that disease or not, such as whether Vibrio cholerae is responsible for having cholera or not. Louis Pasteur developed rabies vaccine. These discoveries are more important, so please make a circle for them or note them down to have a clear idea. Christian Gram, another scientist named Christian Gram, he described the differential staining techniques for bacteria known as Gram staining. So this scientist known as Christian Gram, he found out some techniques in order to differentiate certain level of bacteria. While learning about some bacteria in our microbial world course, we know that certain bacteria has differences in their cell wall structure, and based on them, Gram staining was invented in order to classify that which bacteria has what sort of cell wall structure. This is called Gram staining, and this method was named after the scientist or the inventor scientist Christian Gram. So there are basically 5 scientists or 6 scientists till now we have learned. First one is Louis Pasteur, and Louis Pasteur has versatile discoveries. Then Charles Darwin, theory of evolution. Then Gregor Mendel, the founder of genetics, and he gave us the laws of heredity. Then Robert Koch has significant role in microbiology, postulated, and this is known as Koch or Koch postulate. And of course, Louis Pasteur discovered rabies vaccine, and then Christian Gram. So please keep the flow to learn all the discoveries of these scientists. They are very important. Alexander Fleming. So the most lucky or the luckiest scientist ever in the history of microbiology. He discovered something serendipitously, and I think all of you know the proper meaning of the word serendipity. It is the sudden good luck or the sudden fortune. That means you are doing something in order to have a specific thing or we have another aim, but meanwhile you have discovered something very unique or something very positive for you. So what he was doing, he was culturing a certain type of bacteria in his laboratory, but was failing a lot of time. He was not able to culture those bacteria because in every time he was trying to culture them in a petri dish, every time all the bacteria were lysed or all the bacteria were degraded within that petri dish. After several times, he noticed that there were something like a greenish or bluish type of mold within the petri dish that cause or that is responsible for the degradation of that bacteria. Then the idea struck in him that these sort of things might have some bacteria killing capacity. Later on, he found that this is the mold of the fungus Penicillium notatum. Well, this story will be explained in your another course. However, let me finish it. So, then he declared that Penicillium notatum has a capacity to degrade bacteria. Later on, the American scientists took 7 to 10 years in order to separate Penicillium mold from penicillin, and then it was commercialized as penicillin, or the first ever antibiotic in history. Another Nobel-winning scientist, James Watson, Francis Crick, and Rosalind Franklin, and all thrice of them have won the Nobel Prize in order to, or due to finding or due to discovering the structure of DNA. Watson and Crick find out the structure of DNA, and Franklin discovered the X-ray crystallography procedure. So research led to the discovery of the double helix structure of DNA in 1953. All of them. This is Watson, this is Crick, and this is Rosalind Franklin. Paul Berg, the most unfortunate scientist whose name students will never ever keep in mind. If I ask some students that who is the father or who is the founder of the term recombinant DNA technology, they will answer either Robert Koch or Watson or any other scientist, but not Paul Berg. Let's keep in mind that this is Paul Berg, and this scientist has the credit to discover recombinant DNA technology, the basic tool of today's or modern genetic engineering, through which we have found our modern biotechnology. So, Stanford University scientists who first developed recombinant DNA technology, a method for insertion of genetic material from one organism into another. The technology, or this technology, is the most interesting and the most curiosive technology of all. When I will teach you about how to recombine any DNA with another, or how to ligate or join two different genes, you will know that how beautiful science is, or how beautiful biological science is. There's some recognition about some different types of scientists. You need to get through about them. Now look at this. 1978, Carey Mullis discovers PCR. I shall let you know the details about PCR, but this scientist died in last year. Carey Mullis. Last year or 2020, I can't remember. Last year, 2019. He died last year, and he was a discovering scientist of PCR technology, the most widely used technology in all sort of laboratory all over the world. So we have to know this point about PCR technology. Rest of the technologies you can have a look, and if any of you have any curiosity or any questions regarding this, you can ask me either by email or in our Zoom class. That is all for our first lecture. See you, or we'll have a conversation in our next lecture. Thank you.